Ses. Herkese merhabalar. Mütevelli eğitimimizin değerli başkanı, sayın rektörümüz, rektör yardımcılarımız, dekanlarımız, öğretim üyelerimiz, değerli İstinyeliler, kıymetli misafirlerimiz ve Profesör Doktor Michael Lim. İstinye Üniversitesi Tıp Fakültesi 2024 Bilim Nişanı Takdim Töreni'ne hepiniz hoş geldiniz. Dear Chairman of our Board of Trustees, Rector, Vice Rectors, Deans, Istinye family, guests, and Professor Dr. Lim. Welcome to the 2024 Science Award ceremony hosted by the Istinye University School of Medicine. Stanford Üniversitesi Beyin Cerrahi Anabilim Dalı Başkanı Sayın Profesör Doktor Michael Lim'in bilime kattığı değerli çalışmalarını Bilim Nişanı ile taçlandırmak için bugün buradayız. Programımıza müzik dinletisiyle başlıyoruz. Sahnede Sayın Verda Ferah ve Zeynep Büyükçınar var. Eş zamanlı olarak Doktor Lim'in hayatına dair bazı fotoğrafları da göreceğiz. We are here tonight to honor the outstanding achievements of a scientist with Medal of Science. We will begin tonight's event with music. We have Miss Verda Ferah and Miss Zeynep Büyükçınar on the stage. Simultaneously, we will see the photos of Dr. Lim. Sayın Ferah'a ve Büyük Çınar'a bu eşsiz müzik dinletisi için çok teşekkür ederiz. Thank you very much, Miss Ferah and Miss Büyük Çınar, for the beautiful performance. Şimdi de konuşmalarını yapmak üzere dekanımız Sayın Profesör Doktor Türker Kılıç'ı sahneye davet ediyoruz. I would like to call the Dean of School of Medicine, Professor Türker Kılıç, to give his speech.
Dear Mike, dear Mary, we are very glad to have you here. So it's our honor and pleasure. Welcome. This ceremony witnesses very important firsts, very, very important initiations. And in order to feel and share these initiations more powerfully to you, I would like to talk some parts in Turkish, and in the end, I will end in English as well. I will switch to in the end to English. Bu seremoni benim için çok önemli ve bu seremoninin birçok ilkleri var. Bu ilkleri yaratan kişilere hepinizden biraz sonra teker teker alkış rica edeceğim. Şimdi birincisi bizim mottomuz yenilendi ya da mottomuz artık dizayn edildi. Bu mottomuz okuduğunuz gibi Curiositas et Scientia liberant. Türkçesi şu: merak ve bilim özgürleştirir. Merak ve bilim neyi özgürleştirir? Merak ve bilim bizim zihnimizi özgürleştirir. Bu sadece bizim daha iyi hastalarımıza hizmet vermemizi sağlamaz. Aynı zamanda kurduğumuz bağlarla, kurduğumuz zihinsel bağlarla bizim hayat içerisindeki özgürlüğümüzü arttırır. Dolayısıyla birinci ilk birilerden bir tanesi bu. Diğer slaydı alalım. Pardon, bendeymiş. Bu artık yeni mottomuz. Ve diğer ilk içinde bulunduğumuz salon. Bu içinde bulunduğumuz salonda bir müzik konseri oldu ama konferans olarak bu salonun ilk kullanılması. Bu salon sadece bir kongre merkezi değil, aynı zamanda Muharrem Bey'in vizyonunun bir parçası. Dolayısıyla bu vizyonun bir parçası olarak bu salondaki ilk toplantımızı yapıyoruz. Ben bu vizyonu ortaya koyduğu için ilk önemli alkışı Muharrem Bey için rica ediyorum. Diğer ilklerden biri geçen haftalarda ortaya kondu. İstinye Üniversitemiz ilk kez Times Higher Education Index'te rank edildi ve ilk 1000-1200 arasında yer aldı. Belki biliyorsunuzdur, dünyada 14.000'in üzerinde üniversite var. ve ilk girdiği anda ilk 1000-1200 arasında üniversitemizin girmiş olması çok önemli. Ve bu sıralamada vakıf üniversiteleri içerisinde Türkiye'de 8. sırada, devletle birlikte sıralandığı zaman da ilk 20'de, tıp fakültesi olan vakıf üniversiteleri içerisinde 3. Tıp fakültesi olan hem vakıf hem devlet üniversiteleri arasında da ilk 10 içerisinde. Bu çok önemli bir başarı. Ben bu başarı için çok kıymetli rektörümüzü, Erkan hocamı ve elbette çok kıymetli rektör yardımcılarımızı ve tabii ki en önde cübbeleriyle olan öğretim üyelerimizi, eskiden beri bu tıp fakültesine emek veren herkesi, eski dekanımız dahil, herkese alkışlamanızı rica ediyorum. Tabii ki bu sıralama yeterli değil. Bana göre ikincilik başarısızlıktır. Hedef her zaman birincilik olmalıdır. Bunu da yapmanın yolu araştırma yapmaktan geçiyor. İyi bilim insanı hekim yetiştirmek bizim temel amacımız. İyi hekimler bugünün insanlarını tedavi ederler. İyi bilim insanları geleceğin hastalarını da tedavi ederler. Dolayısıyla önümüzdeki yıldan itibaren 35-40 öğrencimizi %100 burslu olarak iyi bilim insanı hekim olarak yetiştireceğiz. Michael Lim, bilim insanı hekim rol modelliğinin dünyadaki öncülerindendir. Bu nedenle bu tür bilim nişanı törenlerinin amacı bu etkileşimi arttırabilmek ve az önce sözünü ettiğim o merakı ve zihin özgürlüğünü sağlayabilmektir. Biliyorsunuzdur, Nobel alan ya da Nobel'e aday olanların %72'sinin hocası Nobel almış ya da Nobel'e aday olmuş kişilerdir. Yani bilimde de armut dibine düşer. O nedenle bu etkileşmeler, bu rol modellerinin aramızda olması çok önemli. Ve bu sebepten biz özellikle tıp fakültemizin H-indeksi yüksek olan, 20'nin üzerinde olan 32 öğretim üyemiz var. Ben bu 32 kişi için bu kez alkışınızı rica ediyorum. Ve tabii ki öğrenciler. Bu törenin, bizlerin, akademisyenlerin buradaki varlık sebebi sevgili öğrencilerim sizlersiniz. Bu benim sizlerle ilk konuşmam, ilk buluşmam olduğu için çok heyecanlıyım. Ben daha önce 4507 gün dekanlık yaptım ama bu ilk konuşma bambaşka. O nedenle ben hepinizin önce kendinizi alkışlamanızı rica ediyorum. İyi ki varsınız. Bir başka ilk de şu, ben ilk kez kendimden daha küçük birine bilim nişanı veriyorum. Michael Lim'in ne olduğunu, neler yaptığını biraz sonra göreceksiniz. Geçen sene ben kendisiyle tanıştım ve 2001'de benim aldığım ödülü geçen sene Mike aldı. Ve bu da benim için bir başka ilklerden biri. Kendisini bu önemli ödül için bir kez daha kutluyorum. Ve you know, there is always a main hero behind the curtain. So the biggest applause goes to Mrs. Lim Mary. Mary, please stand up. Biliyorsunuz her başarılı kahramanın arkasında ondan daha büyük bir başka kahraman vardır. Mary onların başında. Ben kendisini bu güzel ailesi ve eşine olan destekleri, kendisi de bir dermatolog, çok başarılı bir dermatolog, kendisini kutlarım. Burada olduğunuz için, salonu doldurduğunuz için ve geleceğe daha güvenle bakmamızı sağladığınız için, başka üniversitelerden gelen, cübbeleriyle gelen sevgili hocalarım davetli olarak geldiler. Öğretim üyelerimize ve elbette ki çok sevgili öğrencilerime hepiniz hoş geldiniz. Nice nice konferansları burada gerçekleştireceğiz. Sağ olun.
Sayın Dekanımıza konuşmaları için çok teşekkür ediyoruz. Şimdi ise İstinye Üniversitesi Rektörü Sayın Profesör Doktor Erkan İbiş'i açılış konuşmasını yapması için sahneye davet etmek istiyorum. Now I would like to invite the Rector of Istinye University, Professor Erkan İbiş, to give his opening remarks. Ben teşekkür ederim.
Mütevelliyetimizin değerli başkanı, değerli meslektaşlarım, sevgili öğrenciler, değerli konuklar, çok değerli bugünkü onur konuğumuz Profesör Lim ve çok değerli eşleri. Öncelikle böylesine güzel, önemli bir törende bir araya geldiğimiz için mutluluğumu ifade etmek istiyor ve hepinize hoş geldiniz diyorum. Türker hocamızın nazik ve etkili konuşmasından sonra konuşmak herhalde burada kolay değil. Öncelikle konuşmalarından dolayı kendisine çok teşekkür ediyorum. Tabii teşekkürlerim ileride devam edecek. Ben de heyecanlıyım çünkü bu salonun yapımı aşamasında pek çok geldik. Pek çok kez geldik. Ama içimizde herhalde en çok gelen de 2 günde bir mütevelliyet başkanımız Doktor Muharrem Usta'dır. Salonun tabii hep hayal etmeye çalışıyordum. Nasıl bir şekle bürüneceğini, nasıl olacağını. Ama bugünkü bu görkemli manzarayı görünce gerçekten etkilendiğimi söylemek istiyorum. Ben de buraya ilk defa kürsüsüne çıkıyor, ilk defa bir toplantıya burada katılıyorum. O bağlamda da ben üniversiteye verdiği emekler, katkılar için elbette ki başta mütevelliyet başkanımız olmak üzere herkese de çok teşekkür ediyorum. Şimdi birazdan Profesör Lim'in özgeçmişini dinleyeceğiz. Elbette ki çok başarılı, etkileyici bir özgeçmişi olduğu gerçek. Detayını da hep beraber buradan izleyeceğiz. Ancak benim şöyle hızla bakabildiğim kadarıyla da gerçekten bilim dünyasına çok önemli katkılar sağlamış. Özellikle beyin cerrahisi ya da nöro-onkoloji alanında çok önemli katkılar sağladığını hep özgeçmişinden hepimiz göreceğiz. Ardından özgeçmişinden sonra Lim birikimleriyle, deneyimleriyle, o engin deneyimleriyle bizle paylaşımda bulunacak. O güzel sunusunu hep beraber izleyeceğiz. Tabii ki ben bu tür etkinlikleri çok önemsiyorum. Bir üniversitenin kurumsallaşması açısından çok önemli bulduğumu da ifade etmek istiyorum. Bu törenler, bu ritüeller elbette ki tekrarlanacak. İşte bu kampüsteki bu ihtiyacımız bizim gideren bu salonla beraber pek çok etkinliği de burada sizlerle beraber gerçekleştirmiş olacağız. Bu tören ve bu etkinlik sadece burada kalmamalı diye düşünüyorum. Tabii ki buradan dekanımız Türker Hoca'dan ve Profesör Lim'den benim bir dileğim var. Bundan sonraki süreç içerisinde Istinye Üniversitesi ile Stanford Üniversitesi arasındaki işbirliğini sağlamak, ortak projeleri gerçekleştirmek, öğrencileri, akademisyenleri etkileyen süreçleri teşvik etmek sanıyorum da bu bir sorumluluk olarak da siz üzerinize almış durumdasınız diye düşünüyorum. Elbette kendisine bilim nişanını takdim edeceğiz. O bilim nişanının aynı zamanda bu işbirliğinin de bir sembolü olduğunu, hatta bir mührü olduğunu, damgası olduğunu da buradan da ifade etmek istiyorum. İstinye Üniversitemiz tabii ki çok güzel bilgileri paylaştı Türker Hocam. Ben onları tekrar etmeyeceğim. Gerçekten sessiz, derinden, ağırbaşlı, itibarlı ilerleyiş içerisinde olan bir üniversite. Elbette ki gelecek hedeflerimiz daha da yüksek. Hızla gidiyoruz ama aceleci davranmıyoruz. Yaşımız genç ama olgunlaşma sürecini hızla gerçekleştiren bir üniversite. Dolayısıyla bu özellikleriyle beraber kapsamlı bir üniversite, hem bilimin, hem eğitimin, hem araştırmanın, hem de kültürün, sanatın, sosyalin de olduğu bir üniversite. Ama odaklanmasını da becerebilecek bir üniversite. Bundan sonraki süreçlerde de zaten odaklanarak ilerleyeceğimiz ama bu kapsamlılığı kaybetmeyeceğimiz bir üniversite, çağdaş bir üniversite. Bu bağlamda da üniversitemizin gelecek süreç içerisinde çok daha önlerde, çok daha üst sıralarda göreceğiz. Bunları da sanıyorum şimdi birazdan mütevelliyet başkanımız bunları da size vurgulayacaktır diye belirtmek istiyorum. Ben sözlerimi fazla uzatmak istemiyorum. Öncelikle üniversitemiz adına Profesör Michael Lim'e çok teşekkür ediyorum. Ülkemize geldikleri, üniversitemizi ziyaret ettikleri ve bizi hem konferanslarıyla hem de bu nişanı kabul etmekte onurlandıkları için kendilerine çok teşekkür ediyorum. Tabii ki Türker Hocama teşekkür ediyorum. Çünkü böyle organizasyonları gerçekleştirmenin de kolay olmadığını bilen bir insanım. Arkasında çok emek olduğunu, çaba olduğunu biliyorum. Organizasyonun gerçekleştirilmesindeki çabaları için kendisine de çok teşekkür ediyorum. Sayın Başkanım, tekrar bir kürsüden size teşekkür ediyorum. Çünkü böyle bir imkanı üniversitemize binasıyla, çevresiyle, spor alanlarıyla, kültür, sanat için kullanacak alanlarıyla aslında zenginleşen bir üniversiteyiz. Bir üniversitede herhalde bilimle, eğitimle, kültürle, sanatla zenginleşir. Bu zenginleşmeyi hep beraber yaşayacağız, göreceğiz, izleyeceğiz. Elbette ki buradaki paydaşlar çok önemli. Meslektaşlarımız, öğretim üyelerimiz, akademisyenlerimiz, öğrencilerimiz, çalışanlarımız, herkesin emeği. Dün burada olmayanlar aramızda var, onların emekleri var. Bugün aramızda olanlar var. Bundan sonrakiler var. Ben herkese, üniversitemize dair emekleri, katkıları sadece bu tören bağlamında söylemiyorum. Her konudaki katkılar için de huzurunuzda teşekkür ediyorum. Elbette ki katılımcılara buradan çok teşekkür ediyorum. Öğrencilerimizin ilgiyle katıldığını, araları da doldurduğunu görüyorum. Onlara da teşekkür ediyorum. Hepsine, herkese toplu bir alkışla ben gönderiyorum ve hepinizi saygı ve sevgiyle selamlıyorum.
Sayın Rektörümüze konuşmalar için çok teşekkür ederiz. Thank you Professor Dr. İbiş for your speech. Şimdi mütevelli eğitimimizin değerli başkanı Doktor Muharrem Usta'yı konuşmalarını yapmak üzere sahneye davet ediyoruz.
Bu güzel salonda çok değerli akademisyenlerimizi, idarecilerimizi ve tabii ki tek tek güzide öğrencilerimizi bir araya toplayan Sayın Michael Lim. Öncelikle hoş geldiniz. Dün kendisiyle yaklaşık 2 saat bir arada bulunduk. 2 saatte tabii ki bir insan bir başkasını tanıyamaz. Elbette birbirimizi çok iyi tanıdığımızı söyleyemem. Ancak bunca yıllık kendi tecrübemle şu cümleyi kullanmasam gerçekten üzülürüm. Michael Lim, büyüdükçe küçülen mütevazi benliğinizle sizi çok ama çok içten sevdim. Takdir ettim. O nedenle size çok teşekkür ediyorum. Evet, buradaki ilk toplantımızı yapıyoruz. Tabii ki benim gözümde arkadaki 9 yıl canlanıyor. Geçen 9 yılımız canlanıyor. Salonda benimle birlikte bu yolculuğa başlayan arkadaşlarım var. akademisyen ve idareci arkadaşlarım var. Üniversitenin kuruluşunda Yükseköğretim Kurumu'na sunuma gitmiştik. İstinye Üniversitesi'ni kurmak üzere. Sunumu Türker Hocam yapmıştı. İstinye Üniversitesi'nin takdimine, nasıl bir üniversite olacak diye. Türker Hocam şimdi yolumuz kesişti. Aslında seninle tabii ki uzun yıllardır birlikte devam ediyoruz ama akademik anlamda da bugün yollarımız kesişti. Yeni dekanımız olarak size de hoş geldiniz diyorum. Ve bu arada tabii ki tıp fakültesinde kurucu dekanımız dahil olmak kaydıyla dekanlık görevinde bulunan tüm dekanlarımıza huzurunuzda çok teşekkür ediyorum. ...2025 yılı bizim 10. kuruluş yıl dönümümüz olacak. 23 Nisan'da kurulduk. 23 Nisan 2015 yılında kurulduk. 23 Nisan 2025 10. kuruluş yıl dönümümüz. 23 Nisan'ı olağanüstü coşkuyla kuruluş yıl dönümümüzden bahsediyorum. Olağanüstü coşkuyla kutlamak istiyoruz. Çünkü yeni bir 10 yıla hazırlanıyoruz. Şu anda ekibimizle birlikte, yakında akademisyenlerimiz de dahil olmak kaydıyla, şimdi buradan herhalde öğrencilerimiz de ilk defa duyacaklar. Öğrencilerimizin arasından seçilecek olan 200 öğrencimizi de işin core kadrosuna alarak üniversitemizin bundan sonraki 10 yıllık yol haritasını belirleyeceğiz. Peki 10 yılda nereye geldik? Hani dedim ya, salonda bu yolculuğa birlikte çıktığımız çok değerli arkadaşlarım var. Ne söz verdiysek üniversiteyle ilgili hepsini gerçekleştirdik. İnanmayanlar oluyordu. İnananlar oluyordu. Çünkü çok zor bir iddia, önemli bir iddia ortaya koymuştuk. Türkiye'deki vakıf üniversiteleri arasında 2025 yılında, yani 10. yılı kutladığımızda ilk 5'te olacağız. Uluslararası sıralamada biraz önce Türker Hocam size bahsetti. O sıralamaya İstinye Üniversitesi ilk defa girebildi. Çünkü girebilmenin kriterleri var. Yani biz istesek de önceki yıllarda giremiyorduk. Zannediyorum Türkiye'den bugüne kadar hiçbir üniversite ilk defa girmiş olduğu uluslararası tarafından yapılan sıralamada 1000-1200 sırasından sıralamaya girmemiştir. Muhtemelen bu da bir ilktir. Ama biz gelecek sene, ben şimdi buradan büyük oranda nerede olacağımızla ilgili size bilgi aktarayım. Ya 600-800'de olacağız dünya sıralamasında ama en kötü 800-1000 arasında olacağız. Hatırlarsanız bunlar hep konuşuyorduk. Buralara gidebiliriz diye. Ama bu henüz İstinye Üniversitesi için daha ilk adım. Çok genç bir üniversite. Ya herhalde bir üniversite olabilmek için hani Stanford'ı buradan görüp Yale, Stanford, MIT, Oxford, Cambridge filan. Arkadaşlar 1000 yılı aşkın mazileri olan üniversiteler var. Yüzlerce yıllık üniversitelerden bahsediyoruz. Biz daha 10 yaşındayız. Ya bizim hani üniversite olma yolunda ciddi adım attık diyebilmemiz için bir 50 sene lazım. Onun için şu an varmış olduğumuz yeri çok ama çok önemsiyorum. 50. yılını canlı olarak muhtemelen görmeyeceğiz. Üniversitenin İnşallah görürüz. Kim bilir nerede olacağız ama üzerimize bir sorumluluk düşüyor. Biz üniversitemizi iyi bir yola koyarak, sağlıklı bir zemine oturtarak, yarınlara sağlıklı, düzgün, kaliteli şekilde yolculuk yapabilecek bir üniversite olsun, onu inşa edelim diye uğraşıyoruz. O nedenle değerli arkadaşlarım benim bu sözlerimi şimdi hatırlayacaklardır. Son dönemlerde ısrarla şu cümleleri kullanıyorum. İstinye Üniversitesi madem ki böyle bir hedefe koşuyor, Stanford'dan hocalarımızla işbirliği yapalım. laboratuvarlarla işbirliği yapalım. Harvard'la yapalım. Oxford'la yapalım. Cambridge'de yapalım. Yani Istinye Üniversitesi eğer işbirlikleri, beraber yolculuklar, birlikte çalışmalar yapacaksa dünyadaki ilk 50, ilk 100'e girmiş üniversitelerle bundan sonra nasıl çalışabilirim diye 24 saat kafasını yorsun istiyoruz. Başka şeylere asla yormasın. Bunu asla ve asla istemiyoruz. Peki bu olabilir mi? Dün Michael Lim'le konuşurken ona bir projemden bahsettim. Bir soru sordum. Kendisi de bana bir cevap verdi. Bugün Sayın Rektörümüzle o cevabı paylaştım. Biz 10. yılımızla birlikte yeni 10 yıl için çok ama çok kaliteli doktora öğrencileri, postdoc öğrencilerinden bir grubu dünyanın en iyi üniversitelerine, en iyi laboratuvarlarına gönderelim ve uzun süreli destekleyerek karşılıklı bilimsel know-how paylaşımı, laboratuvar altyapısı, Ar-Ge altyapısı, insan kaynağı karşılıklı değişimi, update olması gibi işlerde yeni bir yolculuğa çıkmamız gerektiğini sizlere söylüyordum. Dün Lim'le konuştuğumda bana, daha doğrusu şu soruyu sordum. Sayın Lim, biz böyle bir şey düşünüyoruz. Hangi düzeyde bu işbirliğini yapsak daha iyi olur? Cevabı şu oldu. Evet, haklısınız, güzel bir yol. Bunu dünyada bilimsel anlamda gelişmekte olan ülkeler zaten yapıyorlar ki onun ülkesi Kore. Kore'yi hatırlayın değerli arkadaşlar. 80'li yıllarda Türkiye ile Kore bilimsel zemin açısından da aşağı yukarı aynı düzeylerdeydi ekonomik olarak da. Ne oldu da Kore bugün çok daha farklı bir yere gelebildi? Elbette burada herkes bunları biliyor. Kendisi beyin cerrahi birimine, bölümüne hem araştırmacı olarak hem klinisyen olarak genç, dinamik, başarma azmi çok yüksek asistan profesörler gönderirsek gönlünü, mekanını, üniversitesini, bölümünü bize açacağını ve bu işbirliğinin kısa dönemli değil, uzun dönemli olacağıyla ilgili iyi niyetini bana dün aktarmış oldu. Tabii ki bunun için kendisine ayrıca teşekkür ediyorum. Buradan şunu söylemek istiyorum. İkinci 10 yılımızda en önemli yapacağımız şey şu. Bunun içine öğrencilerimiz de girebilir başarılı öğrenciler. Biz olağanüstü kaliteli akademisyenlerimizi, bunların mümkünse hepsi üniversitemizden olsun isteriz. Değilse ülkemizden en kaliteli akademisyenlerimizden seçtiklerimizi dünyanın en iyi üniversitelerine göndererek know-how paylaşımı yapmak ve bunu kalıcı uzun bir yolculuğu inşa etmek üzere sürdürmek en öncelikli sorumluluğumuz olacak. Buradan değerli rektörümüze, yöneticilerimize, herkesin huzurunda söz veriyorum ve onlardan rica ediyorum. Ülkemizin kendi alanlarında en iyilerini nerede olursa olsun bularak önce üniversitemizin kadrosunu almak, mümkünse almak, değilse işbirliğini yapmak kaydıyla dünyanın en iyi bilgi üreten yerleriyle işbirliği yapmak üzere yeni bir yolculuğa çıkıyoruz. Bu amacım, bu isteğim sizlere emanettir. Değerli rektörüm, değerli yöneticilerimiz. Lütfen bunu 1. sınıf olağanüstü en iyi şekilde öyle bir yapın ki Türkiye'de gencecik bir üniversite, evet, İstinye Üniversitesi muhteşem bir işe, kalıcı bir yolculuğa çıkmak kaydıyla olağanüstü derinliği olan bir işe imza attı. Herkesin bunu demesini istiyorum. Bunu lütfen sahiplenin. Bana bunu bir daha sormamanızı rica ediyorum. Yetki sizde. Tamamen önünüzü açma konusunda evet buradan söz veriyorum. Diyeceksiniz ki Muharrem Bey ne oldu? Olan hiçbir şey yok. Biz Istinye Üniversitesi'ni daha önce söylediğim gibi üniversitelerden bir üniversite olsun diye asla kurmadık. Böyle bir şeye ihtiyacımız yok. Bu üniversite, sizinle birlikte inşa etmiş olduğumuz Istinye Üniversitesi, önümüzdeki yıllarda gencecik arkadaşlarımızın, bizden mezun olacak gencecik öğrenci arkadaşlarımızın, iyi ki bu üniversiteden mezun oldum, şahane bir üniversite oldu, muhteşem bir üniversite oldu, üniversitemle gurur duyuyorum, her yıl başarısını gördükçe olağanüstü mutlu oluyorum, iyi ki İstinyeliyim diyeceği bir geleceği onlara armağan etmek zorundayız. Hiçbir çocuğumuz, hiçbir evladımız İstinye Üniversitesi'nden sıradan bir üniversiteden mezun olduğum gibi bir duyguyla, bir gelecekle mezun olmasını asla ve asla istemiyoruz. Ve şimdi sevgili öğrencilere biraz önce söylediğim seçilecek olan 200 öğrenciyle ilgili bir bilgi aktarmak istiyorum. Değerli gençler, 200 öğrencilik bir grup oluşturulacak. Bu 200 öğrenciyle nasıl bir amacımız olacak? O 200 öğrenci buradaki 12.000 öğrencinin temsilcisi olacaklar bir anlamda. Bu ne demek? Öyle 200 öğrenci ki entelektüel herhangi bir konuşulduğunda, gündeme geldiğinde üniversiteler arasında Türkiye'deki en iyi olduğunu iddia eden üniversitelerin öğrencileri arasında kendilerini birinci sıraya koyacak kadar bilgi ve birikim sahibi olacaklar. Yapay zeka konuşuluyorsa en ön sırada onlar olacak. Ekonomi konuşuluyorsa onlar olacak. Ama bu 200 öğrencinin en önemli özelliği şu olacak. Biz onlarla, ben de vakit ayıracağım öğrencilere. O öğrencilerle biz bir konu üzerinde çalışacağız. En çok da bir konu üzerinde. Eleştirel düşünce ne demektir? Eleştirel düşüncenin varabileceği noktalar nelerdir? Bunu yakaladığında bir insan nelere, nasıl yolculuk yapabilir? Analitik bakış nasıl bir şeydir? Analiz ve sentez yapabilmek ne demektir? Özetle öyle bir grup oluşturmak istiyoruz ki o grup bütün bu bilgilerle donanımlı olsun ve üniversitenin içerisindeki binlerce öğrencinin de elçisi olsun. Yani demem o ki 10.000, 12.000, 13.000 öğrencinin hepsini biz olağanüstü kaliteli şekilde yetiştiremeyebiliriz. Ama içinizden 200 öğrenciyi biz çok donanımlı hale getirdiğimizde onlar kendi arkadaş çevrelerinden 2.000 kişiyi farklı hale getirebilir. O 2.000 kişi mahallesindeki 20.000 kişiyi farklı hale getirebilir. O mahalledeki 20.000 kişi çevrelerini değiştirebilir. Yani İstinye Üniversitesi topluma katkı anlamında da çok kaliteli elçiler, gencecik öğrenciler yetiştirsin istiyoruz. Sizlerle ileride yolumuz kesişecek değerli öğrenciler. Biz sizlerin gelecekte olağanüstü işlere imza atan, evrensel ölçekte kendini ispat edebilmiş öğrenciler olmasını istiyoruz. Ve sizin için varız. Tüm kadrolarınız, tüm hocalarınız, bizler, gencecik yaşlarında olan sizler için varız. Evet, gerçekten sizler için varız. Bugün burada konuğumuz olan Michael Lim sizin için olağanüstü bir örnektir. Lütfen onun da hayatını inceleyin. Bu kadar mütevazi, nazik, kibar, beyefendi ama Stanford'ın beyin cerrahisi Anabilim Dalı Başkanı olan bir kişiden bahsediyorum. Nasıl olunabiliyor? Hayatınızda hep kaliteli, derinliği yüksek, dünyaya katkı sağlayan, hayata dair amacı olan ve imzası olanları örnek almaya çalışın. İşte Michael Lim burada. Biliyorsunuz ben konuşmaya başladım, devam eder giderim. Ama benden öte konuşması gereken misafirimiz burada. O nedenle sözlerimi noktalıyorum. Hepinize çok çok teşekkür ediyorum. sevgilerimi, saygılarımı sunuyorum. Sağ olun.
Mütevelli heyetimizin değerli başkanı Doktor Muharrem Usta'ya konuşmalar için çok teşekkür ediyoruz. Thank you Dr. Usta for your speech. Ve şimdi sizlere onur konuğumuzu kısaca tanıtmak istiyoruz. Profesör Doktor Michael Lim, Stanford Üniversitesi Beyin ve Sinir Cerrahisi Anabilim Dalı Başkanı ve beyin tümörleri ve trijeminal nevralji konusunda uzmanlaşmış, kurul onaylı bir beyin cerrahıdır. Johns Hopkins'te geçirdiği süre boyunca Doktor Lim, en büyük beyin tümörü ve trijeminal nevralji muayenehanelerinden birini kurdu ve hastaları için en gelişmiş cerrahi teknolojileri ve teknikleri kullandı. Hasta deneyimini önceliği haline getirmiş olarak meslektaşları ve hastaları tarafından dürüstlüğü ve şefkatli bakımı nedeniyle takdir edildi ve Health Network Foundation'dan bir hizmet mükemmelliği ödülüne layık görüldü. Bir mentor olarak Johns Hopkins Üniversitesi Tıp Fakültesi tarafından seçkin bir öğretmen olarak onurlandırılmak da dahil olmak üzere çok sayıda öğretim ödülü kazanmıştır. Amerika Birleşik Devletleri ve dünya genelinde beyin cerrahisi ve onkoloji eğitiminin şekillendirilmesinde aktif olarak yer almaktadır. Nöro-onkoloji alanında prestijli bir ödül olan 2023 Abhijit Goa ödülünün sahibidir. Doktor Lim'in araştırma ilgi alanları kanserle savaşmak için bağışıklık sistemini kullanmaya odaklanmıştır. Laboratuvarı kanser hücrelerinin bağışıklık sisteminden kaçış mekanizmalarını anlamaya odaklanmıştır. Bulgularını laboratuvardan kliniklere başarıyla aktarmış ve beyin tümörleri için birkaç büyük ulusal immünoterapi klinik denemesi yürütmüş, ayrıca yönetmiştir. Doktor Lim'in bibliografyasında glioblastoma için immünoterapi, stereotaktik radyasyonla tedavi edilen glioma hastalarının uzun vadeli sağ kalımı ve nöropatik ağrının tedavisi gibi konularda 300'den fazla makale bulunmaktadır. Çalışmaları Science Translational Medicine, Clinical Cancer Research, Lancet Oncology, Nature Immunology ve daha birçok yayında yer almıştır. Ayrıca 20 kitap bölümü ve monografi yazmıştır. Doktor Lim, beyin tümörleri için immünoterapi alanında bir dünya lideridir. Dünya çapında konferans ve seminerler vermek üzere davet edilmesinin yanı sıra Amerikan Klinik Onkologlar Derneği, Amerikan Nörolojik Cerrahlar Akademisi, Kuzey Amerika Radyoloji Derneği, Beyin ve Omurga Metastazları Yıllık Sempozyumu, Nörolojik Cerrahlar Kongresi ve diğer toplantılarda beyin tümörleri için immünoterapi, beyin cerrahisi teknikleri ve beyin tümörlerinin yönetimi konularında platform sunumları yapmıştır. Ayrıca Amerikan Klinik Onkoloji Derneği konferansında CNS oturumunun platform başkanı olarak görev almıştır. Doktor Lim, Amerikan Klinik Onkoloji Derneği, Nörolojik Cerrahlar Kongresi, Amerikan Nörolojik Cerrahlar Derneği ve Nöro-Onkoloji Derneği üyesidir. Doktor Lim, Nöro-Onkoloji Derneği'nin ve Amerikan Klinik Onkoloji Derneği'nin CNS bölümünün program eş başkanı olarak görev yapmıştır. Ayrıca Amerikan Nörolojik Cerrahlar Derneği ve Nörolojik Cerrahlar Kongresi Ortak Tümör Bölümü Yürütme Komitesi de dahil olmak üzere birçok yürütme komitesinde görev almıştır. Saygıdeğer konuklarımız, şimdi Bilim Nişanı Takdim törenimize geçmek istiyoruz. Sahneye Sayın Rektörümüz Profesör Doktor Erkan İbiş'i, Rektör Yardımcılarımız Profesör Doktor Peyami Çelikcan ve Profesör Doktor Hatice Gülen'i, Sayın Dekanımız Profesör Doktor Türker Kılıç'ı, Dekan Yardımcılarımız Profesör Doktor Nuriye Taşdelen Fışkın'ı, Profesör Doktor Ozan Özkaya'yı ve Profesör Doktor Hikmet Koçak'ı davet ediyoruz. Şimdi tüm değerli misafirlerimizi, değerli konuğumuzu selamlamak üzere ayağa kalkmaya davet ediyoruz. Değerli konuklarımız, bu yılın Istinye Üniversitesi Bilim Nişanı'nın sahibi Profesör Doktor Michael Lim'i sahneye davet ediyoruz. Teşekkürler hocalarım. Pointer.
Okay. So great. Well, thank you so much for this incredible honor. I am so honored and privileged to be here today. Want to, first of all, I think I had the same picture as Dr. Kilic. Want to first say thank you so much for having me here today. I've had an amazing few days here today. And I've got to know a lot of the people here at Istinye University, and I got to speak again with Mr. Usta. I got to spend time with Begum Usta and Gulsa. Thank you so much for taking me all around. And Dr. Kilic, thank you so much for this invitation. I've been so honored. And Ikam, I also want to say thank you for taking my wife and I around, but also to the distinguished leaders of this university. I couldn't be more excited to be here. As I've gotten to know this university, as you said, it's 9 years old, but as I think about this university, I think of it as this place full of bright students. You get some of the smartest minds in the country and in the world here. And I get this sense of you have no limits. I get the sense that you have this boundless energy, and the only place you're going to go is up. And I think in some ways you're going to create your own traditions to make this an amazing and great place. And as Mr. Usta said, I am excited and I'm committed to working with you, because I think that you will be, you are already, but you'll continue to do great things. And I really am excited to collaborate with you as well as our university. So again, thank you all for showing me this incredible hospitality. Thank you for showing my wife this incredible hospitality. And I think symbolically, if you think about this historically, Istanbul has been the center really of the world. And I think that there's been so many great things, and I can see that this is the next great thing that's going to continue to change the world. So thank you again. So today, okay, is that better? Yeah. So today, as I reflected on what I was going to talk about, I think all of us realize that life is short and that it's a precious thing. And so as we go through life, it's a journey. And everyone in this room is at different stages in their journey. Some are just beginning as students. Others here are professors mentoring students. Others are in leadership, bringing up the next generation. And I think we're all going towards one common goal, which is to make life better for humankind. But as I reflect back in my own life, I think that each of you will start, especially from the students, on your own journey in life. And I couldn't be more excited for you because I think it couldn't be a better time in medicine to start this journey. And I'll talk to you a little bit about some of the work I've done, and then talk to you about what I think is going to be the future. So, as Gilso said, I did my medical school at Hopkins, and then I did my residency at Stanford. And my dream, it wasn't to be a chair, actually, it was to be a physician scientist. And I wanted to be a physician scientist studying cancer, particularly brain cancer. And when I finished my residency at Stanford, I had the opportunity to go back to work at Johns Hopkins. On the left, or on the right actually, is my former chair, my boss, from when I was in medical school, and when I was a faculty member, Dr. Henry Brehm. And he gave me this offer letter, and I signed this offer letter in a very historic room at Johns Hopkins. In that room has furniture of Harvey Cushing, William Halsted, people who defined modern medicine. And he said that you're going to be able to make a difference and change the future. And he gave me the opportunity to do that. But to start your journey, you need to come up with a vision. You've got to think about what you want to do to change the world. What is it that you want to do to affect people positively? And so this was the vision I presented to Dr. Brehm at the time. I said I wanted to create a brain tumor immunotherapy program. And the brain tumor immunotherapy program would have a basic science component, a clinical trials component, and a translational component. You had to have all three in order to push the field forward for a difficult disease such as glioblastoma. And I have to say, when I started my career as a physician scientist, I struggled in the beginning. And I think struggles are so important in making you figure out where you want to go. And in the beginning, I didn't know what I was going to focus on. And I tried different things. I tried things such as STAT3 pathways, and other IL-17. But it was important that you struggled. But before I started as a physician scientist, I thought I met my goal because I had been given a lab, I had personnel and protected time, but it was really a time of struggle, as I mentioned. And it's when you struggle that you realize that you have choices and forks in the road. And I think it's through struggles that you continue to push forward. And so one of the things that's important to understand is that when you fight cancer, it's a very difficult disease. It's not something that's straightforward. And one of the things that I think is important to understand is this is the tumor that I dedicated my life to trying to fight. As you can see on the bottom right scan of this person's head was a brain tumor. And this is a young woman who came to me just bumping into the wall while she was a waitress. And her life changed overnight because after I took out her tumor, the diagnosis came back as something called glioblastoma. And glioblastoma is one of the most aggressive types of tumors. And as you can see, I did surgery. It went really well. It looked like there was no residual tumor. But even going through chemo and radiation, a year and 2 months later, her tumor started coming back. And even though I went back and took all of it out again, her tumor came back 6 months later. And I remember her having this conversation with me saying, so it sounds like we've run out of options. And I remember saying to her, I wish I had more to give you. And she ended up passing away 3 months later from this cancer. And so if you look, this blue line here is the survival, the radiotherapy plus temozolomide. I guess the laser's not working. But if you look at that survival, we're still not doing very well. 50 years of research, and we still haven't done well for these patients. So we need to figure out a better way to take care of these patients. We got to come up with new therapies. And we realized that just cleaning out that tumor and getting all of that abnormality out on the MRI wasn't going to cure somebody. And so we needed to come up with a therapy that could potentially help people. So as I mentioned before, throughout my struggles and seeing patients die through this experience, one of the things that I value as a physician scientist is that we can learn from our patients every day. And so in this case, this was a patient that came into my clinic about 3 years into my practice. And he was this gentleman who had a solitary brain met, you can see that there with the red arrow, from kidney cancer. And he was on a clinical trial of a drug I'd never heard of. It was called MDX-110. And I remember telling this gentleman, you need to have surgery, because if we take this tumor out, you can live longer and you could have a better quality of life. So we took out his surgery. We took out his tumor and we didn't have any problems from surgery. But the pathology report came back as no cancer. All those spots you see on that scan on the H&E and the CD8 are all inflammatory cells. They're all immune cells there. It wasn't any cancer cells. And as I watched this gentleman over the next 4 months, he had a complete response. All of his cancer disappeared. This was a gentleman who had great stage 4 kidney cancer, was told he was going to live probably less than a year. All of a sudden was cured and now had a full life. And that clinical trial was on a drug called anti-PD-1. It was actually, I saw one of the very first patients ever treated with this drug. And you might have heard these commercials today because it's now everywhere. It's called Keytruda or Opdivo. I'm sure you've seen the commercials throughout the world for this. But this drug, we had no idea what we were dealing with. And what it is that this drug interacts with the second signal on the T cell. So when a T cell binds its antigen, it has a second signal that tells it to turn on or turn off. And you have that normal signal to turn cells off to prevent autoimmune diseases in your body. Otherwise, you develop things like rheumatoid arthritis. But cancer's also figured out to take that signal to turn immune cells off. So if you blocked it, all of a sudden, immune cells were killing cancer cells. And having seen that, over the ensuing months, I got to see people with melanoma, lung cancer, kidney cancer, all getting cured from this. And as you can see, the seminal studies were done at Johns Hopkins when I was there at the time. This was going to be a drug. This was, I realized, a revolution in cancer care. And so having the brain tumor immunotherapy program and the vision, I said, we need to have that drug. And we collaborated with Dr. Drew Pardoll at Hopkins, who was my mentor when I was a medical student. Drew Pardoll and Chuck Drake gave me anti-PD-1. And so we went and we tested in the animal models. And Jing Zhang, who was a postdoc with me at the time, and now is a full professor at the University of Washington, gave mice with glioblastoma anti-PD-1. And there's no way to get a laser. The laser's not working. Okay. If you can see on the middle curve there, when the animals were treated with anti-PD-1, they did better. They survived longer. And in fact, if you gave it with radiation, they did even better. And so we were able to publish that. And you can see that project started in 2010 and completed in 2012. And a few years later, there were even case reports of patients getting anti-PD-1 with glioblastoma getting responses. So having generated that data, I remember in 2012 going to a company called Bristol Myers Squibb, which was making that drug, and saying, can you give this for our glioblastoma patients? These patients need it very badly. And at that time, Bristol Myers Squibb actually said no. They said, we have other programs going right now. We do not have the bandwidth for glioblastoma. But in 2013, the leadership changed in BMS, and that's the importance of persistence. You're going to be told no a lot in your career, and you have to keep going back and asking. And this is to advocate for your patients. So I went back in 2013, and there was new leadership at Bristol Myers Squibb. Vlad Korich, Karina Tate, and Robert Latek at Bristol Myers Squibb came and they said, we absolutely need to do this for glioblastoma. And I remember saying, great, can we have some of it for a phase 1 trial? And they said, no, we're going to go for 3 phase 3 clinical trials. And so it was an exciting time because I remember there was a group of us from several institutions across the country, and we all met up in New York City and designed the phase 3 clinical trials. 1 in recurrent glioblastoma and in 2 for the newly diagnosed glioblastoma. There are 2 subtypes, molecular subtypes, MGMT methylated and MGMT unmethylated. And so, as you can see from the timeline, from 2014 to 2022, we ran 3 large phase 3 clinical trials. But the results of these clinical trials came back very different. There wasn't improved survival. I remember when we looked at this data, we were all heartbroken because we thought we found something for our patients with glioblastoma, but it didn't work. After thousands of patients later, we realized that this wasn't going to work. And the field of immunotherapy had matured over those 10 years, and we realized that not every cancer was going to respond. Prostate cancer, pancreatic cancer, and glioblastoma, most notably, did not respond to checkpoint inhibitors. And I remember being very heartbroken and taking a step back. But this is where persistence is important. And this is where being a clinician scientist, I think, is a strength. So we took that loss and we said, what are we going to do? Well, let's go back and let's study the cancer again. And what we realized is that all cancers, not all cancers fight or hide from the immune system the same way. And glioblastoma has different mechanisms to hide from the immune system. So one of the things that we did was when we went back, we started to realize that the immune cells that went into glioblastoma, particularly the CD8s or the lymphocytes, were exhausted. And this was a new term that I learned, exhaustion. I said, what's exhaustion? So that is actually a term that's well known in infectious disease. When you have chronic hepatitis, your T cells become exhausted. It turns out that if you constantly stimulate your T cells, the T cells lose their ability to kill, and that is exhaustion. And it turned out that Magda, who's one of my postdocs, and then Dr. Peter Fecci's lab also showed that when you pulled out those T cells from glioblastomas, they couldn't kill, and they were exhausted. So we said, okay, this is probably a barrier for our patients with glioblastoma in getting an immune response. So we said, okay, we need to understand this better. And so we went from a phase 3 clinical trial and observation, and we said we need to go back and understand what's going on. So we wanted to understand the mechanism behind exhaustion. What cells are driving this exhaustion? And is this exhaustion permanent? Back in 2015, we didn't have the sophisticated science that we have in 2020. So I'll talk about kind of how we got deeper into this question on how to make things better for glioblastoma. So what we did was, when we went back and we looked at the exhausted T cells, one of the things that we noticed was that these T cells were expressing another checkpoint molecule called LAG3. So it wasn't PD-1, but this other molecule called LAG3. And what we did was we went back into the lab and tested that question. So Sarah Harris-Bookman, who was a technician in my lab, and Demetrius Matthews, said, let's see what happens when we manipulate this. So first of all, what they did was we went and looked at a whole bunch of patients who had glioblastoma. And if you look on the top right, all those little brown dots are T cells that are expressing LAG3. It turns out that almost 100% of patients with GBMs have LAG3 positive T cells. And it turns out that if you look in the middle figure, if these T cells have LAG3 and PD-1 expressed on them, they do not have the ability to kill. They're exhausted. That y-axis is something called interferon gamma. That's a marker for killing. But if the PD-1 and LAG3 is blocked on the very right, you can see all of a sudden 40 to 60% of the T cells were able to kill again. And then what they did was they treated the mice with anti-LAG3 and anti-PD-1, and it's that purple line there. All of a sudden, they were working synergistically, and these mice were getting very nice responses to anti-PD-1 and anti-LAG3. The black line is a knockout mouse for anti-LAG3, so it worked even better. So what we did was we went back to BMS, Bristol Myers Squibb, and said, can we run another trial? They said, we don't have the resources, i.e. we don't have the money to fund the trial right now, but we'll give you the drug if you can find another source. So this time around, we had to go to the National Cancer Institute in the US. So the NIH, or the National Cancer Institute, we applied and we got this trial funded, where we were going to give patients either the anti-LAG3 alone or anti-LAG3 plus anti-PD-1 in recurrent glioblastomas, okay? And so this time around, if you can look up on the top left panel, that's a patient who had a glioblastoma in the beginning and then had it removed. And despite the chemo and radiation, you can see it says recurrence. So then he went into our trial. And you can see after he had the surgery, initially it looked clean, but on the bottom row there, that's cycle 3, 4, and 5, you can see that it looked like the tumor was coming back. And in fact, we were actually discouraged by that scan. But he was also developing thyroiditis. He had a little bit of autoimmune disorder going on. And this person is now alive at almost 60 months. He's 5 years into this, and he's alive. When you have a recurrent glioblastoma, the average survival is 6 months, okay? We said, okay, well, that's one person. Then we had another patient. This second patient, as you can see, had 2 spots. You can see the red line and the green dot up there. And over time, that person's tumor started melting away. And as you can see, it didn't cure everybody, but in this person's tumor, it slowed it down enough so that this person lived almost 3 years. And yet another person had their tumor melt away. So all of a sudden, here's another one that had a nice response. We saw people doing well on this therapy. And the blue line is the historical control, and the red line is the treatment arm. And as you can see, at 12 months, 54% of the patients were alive, okay? And now they're close to 60 months, almost 3 of those patients are alive. So we also ran a concurrent arm, I'm not showing that slide today, but where we gave patients LAG3 alone, and again, it was close to the blue line. So statistically, we were very encouraged that this therapy might work. And then what we did was we went back to the laboratory again, and we started looking at the samples of these patients. And it showed, for example, in these responders, the T cells were responding, and they were vigorously expanding once they got exposed to the LAG3 PD-1, our immunotherapy. And when we started looking and doing genetic analyses, they had this, what they call interferon signature. It's a signature that shows inflammation that's going on. And they had activities such as antigen presentation, showing that, again, the immune system was revving up in these patients. And again, it correlated beautifully. So with that, we then went back to the NCI and said, okay, we did a phase 1 trial of only 24 patients. We need to now do this in a bigger trial to test efficacy in a true randomized fashion. And so to do that, now we're going to take patients with glioblastoma and we're going to treat them either with anti-PD-1 and anti-LAG3 or standard of care, which is the CCNU. And now we're going to treat 170 patients to see if this will work, okay? And so if you look at this, this is, I should say 10, but this is a 10-year journey and counting. Around 2015, 2016, we had an idea that maybe the PD-1 alone wasn't working. So we went back to the laboratory, identified a new target, and then tested it in an animal, and then went to a phase 1 clinical trial to make sure the drug was safe. But then we saw activity. We saw cancers melting away. And now we have this trial open. It's been open for 2 months across 600 sites in the United States to recruit these patients. And so again, you can see how this was a journey going back and forth from patients to translational studies and back. But on the other hand, the thing that bothered us also was that we still didn't understand the mechanism behind exhaustion. Why? Why is it that these T cells can't kill? Okay, so what we did was we said, we got to understand this phenomenon better. It's not just that this LAG3 protein is elevated and blocking it will make it better. Still, if you can see, about 80% or 70% of those patients still passed away. So there must be another explanation to this. And so as we started looking across the literature, other people verified this exhaustion in other cancers too. So it's not just a problem in glioblastoma, but this red line is something called an exhaustion score. And if these T cells are constantly stimulated, they become exhausted. So Christopher Jackson, who was a resident in my lab and now on faculty at Johns Hopkins, came up with a really neat idea. He said, what if we take cancers that respond to immunotherapy and cancers that don't respond to immunotherapy from patients, and then we pull out the T cells. And then we sequence them and look for differences, and maybe we'll figure out a gene that explains why the exhaustion happens. And so what we did was we took bladder cancer and kidney cancer, which responds to immunotherapy, and we took prostate cancer and glioblastoma, which don't. And this is a lot of bioinformatics. It took almost a year for our bioinformatics people to sort through this because it's terabytes of data. But after we went through, we found two genes, something called metronome, that's up at the top right, and CXCR6. Now, I won't talk about metronome, CXCR6, because we got scooped on that, but it validated our findings. But metronome was this protein that we thought was very interesting. Now, metronome hasn't ever been described in cancer before. It was only described in exercise physiology. It turns out that this protein is responsible for metabolism and helping you stay warm, or especially kids stay warm by turning white fat into brown fat. It's a signal for that. But there was literature that said if you created these knockout mice, these mice developed really bad sepsis. They had really vigorous immune responses. So we said, well, maybe there's something to this. So we started with very simple assays. We said, okay, is this protein really there in glioblastoma? And sure enough, when we just started looking at glioblastoma, it was there. And then we went to our mouse level, our mouse studies, to say we have to make sure this is relevant. In fact, the exhausted T cells were expressing this metronome protein. And on the right, if they were exhausted, they had less interferon gamma, which meant that they were essentially not killing. And so we said, okay, well, it seems like this protein correlates with exhaustion. So then what we did was we started manipulating the T cells. And when we started treating the T cells with repeated stimulation, you can see on the right, the metronome levels started going up very highly, sky high, actually. And then what we did was we took known literature from our colleagues who have CAR T cells. And in the patients who the CAR T cells didn't work for, they had elevated levels of this metronome protein. So we said, okay, there's probably something to explain that. So we said, okay, there's this gene, it seems to correlate. How is it causing exhaustion? So what we did was we found in the gene studies that there were a lot of genes that were related to metabolism in the body. And so what we did was we first started looking through what's called a seahorse assay. And we started noticing that when you gave metronome to these cell lines, they were essentially going into glycolysis. Now, I don't know if you remember back to your high school biology days, but you remember when you take a sugar molecule and it gets broken down, it can be broken down through glycolysis, which gives you only 2 ATPs, 2 units of energy, or it can be processed through what's called the Krebs cycle. The Krebs cycle gives you 36 ATPs, or 36 units of energy, okay? And so the 2 ATP glycolysis is a highly inefficient way of getting energy. But it looked like it was making the T cells who lived off 36 ATPs live off 2 ATPs. It was essentially making those cells breathe through a straw. You took away most of their energy. So we said, okay, this might be something related to energy. So exhaustion just isn't a switch, but in fact, exhaustion is an energy problem for the T cells, okay? And so what we realized is that there's probably a ratio between glycolysis and oxidative phosphorylation. and probably that happens in the mitochondria. And so what we did was we actually first started looking at the mitochondria of these T cells. And I don't know if you can see, but in the middle panel, it says untreated. The mitochondria looks like a little jelly bean there with little sharp lines. That's what your mitochondria of a normal T cell is. That's a normal, healthy organelle. But on the right, when you gave metronil, those little jelly beans blew up like a balloon, and all the insides look scrambled up. In other words, the mitochondria stopped looking healthy. Phenotypically, those mitochondria were being altered, and they weren't, we think, weren't working. So we said, okay, we took a look at this under a microscope. They look different. However, is the energy changing? And so then we measured the depolarization in the mitochondria and measured reactive oxygen species. And it indeed, when these T cells were exposed to that metronil protein, all of a sudden you had increased reactive oxygen species and you lost what's called the gradient. And so that explained why they couldn't go through the Krebs cycle. The T cell, the mitochondria, which is your powerhouse of the T cells, were not working. And so when we started doing a little bit more sophisticated metabolomics through seahorse assays, we basically realized that there was metabolic reprogramming happening in exhausted T cells. And so cancer was causing these T cells to not have energy. And so when we looked at something called spare respiratory capacity, it just means your ability to, think of it as almost like your ability to breathe. The red line with the metronil, those T cells also lost their ability to have any spare respiration. But if you created a knockout mouse, basically you took out the metronil, you could preserve that spare respiratory capacity, and the ATP production or the energy production went back up. And it turns out that if you left these T cells exposed to metronil over a long time, they basically then got stuck in glycolysis. And they could live for a little while on glycolysis, and then they die off because they only got 1/18 the energy that they needed to survive or kill. So then what we did was we had mice, and we found a knockout model. And when you knocked out the metronil in those mice, it actually worked well for glioblastoma, prostate cancer, and even colon cancer. All of a sudden, these animals were starting to live longer because their T cells were working. And it turns out that when you look at the metronil protein, it's actually working through a pathway called PPAR delta. And so when metronil is exposed, when the T cells are exposed to this protein, this pathway goes up and then causes the mitochondria to get disrupted. So what we've been able to do is we went back and we said, why are the patients with anti-PD-1 who got treated with anti-PD-1 not working? And we realized that it's a T cell exhaustion. And to understand that, we realized that there's probably cytokines like this metronil that get secreted, and when the T cells get exposed to that, they activate this PPAR delta pathway that ultimately causes the mitochondria to shut down, and the T cells lose energy, and they can't kill. It's like basically taking your electric car and they're pulling the battery out of the electric car. The electric car won't work, the T cells won't work. And so we're excited about this because now we have a potential opportunity to come up with therapeutics, and we're coming up with new ideas, and we have some antibodies that we've created now to counteract that. But then we realized that this was an important paradigm to understand. It isn't just the T cells, but there's actually metabolic reprogramming that's going on in the entire tumor. And it turns out that as you looked, and I'm not going to talk about that today, we started looking systematically. So we looked at the CD8 cells, we looked at regulatory T cells, which are T cells that are meant to suppress, and cancers like using that. We looked at myeloid cells like macrophages and dendritic cells, and we looked at cancer cells. And what we figured out was that cancer, through the Warburg effect, likes to push everything into glycolysis. And when it pushes things into glycolysis, the CD8 cells work, the macrophages actually adopt a phenotype that promote cancer growth. Cancer cells actually are going through glycolysis, and they're creating a lot of this neurotransmitter called glutamate, which actually helps cancers grow. And these Tregs are thriving. But if you can push the microenvironment back into oxidative phosphorylation, all of a sudden you can get these immune cells to start killing cancer again. So I tell people, if you think of T cells like plants, if you plant them in the Amazon rainforest where there's lots of rain, it's oxygen rich, they'll grow like crazy. But if you take those plants and try to plant them in the Sahara Desert, which is what glycolysis is, they're not going to grow. And you can keep putting more and more plants there, but they're not going to grow. And so the only way we think now, this has changed the direction of our lab, is if the Sahara Desert is what you're left with, then we got to bring topsoil there, you got to bring irrigation there, and you got to probably bring some shade to so that the plants aren't melting under the sun. And so, for example, John Choi and Lily Kim are now actually altering the tumor microenvironment by giving an oxygen-rich environment in hopes of making these cells, putting these cells in the best situation to survive. And this field is actually moving forward very quickly. This is a very neat paper. It's not our paper. But if you look on the top left, it's an electron micrograph of a cancer cell. And the cancer cells actually have spindles that are sticking out, kind of like a spider web, okay? And when the T cells come by, they catch the T cells through that spindle. They're called nanotubules. And when they do catch them, they grab onto them, and then they reform their nanotubules and poke in that middle image, poke into the T cells. And then when they poke into the T cells, they actually start sucking out the mitochondria, kind of like they're sucking out, I don't know if you ever had boba or bubble tea, but they're sucking up the little bubbles of mitochondria and bringing them into the cancer cell. It's a striking video, but they're basically taking out the mitochondria to make those T cells weak. On the bottom side, there was a very neat study in something called Neuron, where they showed that nerves, if you have nerve damage, need mitochondria to heal. And they showed that when you cause nerve damage, macrophages come in and inject mitochondria into your nerves to help you heal. And if you block that injection, the nerves don't heal. So the mitochondria are essentially currency, and energy is probably central to many of these processes. So with that pathway, again, it's probably energy driven, and that's probably the mechanism behind exhaustion. And so initially, as you can see, the results for the glioblastoma studies have been very disappointing for us. But being a physician scientist, you have the ability to go back into the lab and ask questions at multiple levels. And we realized, one, we find targets and we can target them directly, but there's also opportunities to understand mechanism, why something is happening. And to us, we figured out that there's another component of metabolism that causes this exhaustion. And we now are developing new sets of therapeutics for the patients in hopes of helping our patients with glioblastoma. Okay, so those are two stories that I wanted to talk to you a little bit about science, but I also wanted to talk to you about impact. When I started my career, and as I said, I was struggling a bit, I realized that when we did the first PD-1 study, we created a lab where we could test agents quickly and then decide whether or not to move to clinical trials. And so from the initial studies, you can see that we were able to launch a phase 3 clinical trial. We developed intellectual property, and we were able to mentor and train a lot of great new generation of scientists from the lab. But we also created this infrastructure, and this translational infrastructure came from the fact that we could go back to the bench. And with that, as we went back to the bench, we could identify new targets, and then we could test them in the animals. And as a result, from our brain tumor immunotherapy program, these are all examples of students and postdocs in my lab who published the various sets of paper that then translated into clinical trials. So we haven't had just two, but we've had a lot of clinical trials come out, and we're able to go back and forth to help patients. And that's been a very gratifying experience for me. And as I said before, this is not something that happens overnight. These are journeys that take about 10 years to do, to go back and forth in the lab. But as I said, as a clinician scientist, you get to learn so much, and you can really help push the field forward. But this doesn't happen alone. And when I started, and Dr. Brem hired me, I started with one technician, and that was it, and some seed money. But as our program grew, it's important to work as a team. And as you can see, the radiation oncologist, the medical oncologist, neuropathologist, neuroradiology, even my wife, and clinical trials division, as well as the basic scientists, and even the legal team at Johns Hopkins had to be involved because we brought in so many people. That's the only way to move forward. You have to be collaborative. And with that, as we built the lab, we worked with many companies. And some people, I have to say, I have been so encouraged by working with industry. There are people in industry that truly want to see us cure glioblastoma. And these folks at these companies came and worked with us in our laboratories to try to test their reagents. And then we helped them either bring it to clinical trials or avoid clinical trials for them. But as I've gone through this, and now my role as a chair, I tell people I'm coming back to the idea of stages in life. And I'm sure some of you have seen the Taylor Swift concerts. Yes? And it's called the Eras Tour. In the US, there's something called ERAS, which is for the match, the residency match. And I foolishly thought that it was ERAS when I first saw the title, but it's Eras. And if you listen to her music and you listen to her concert, she sings 44 songs. And in each of the songs, she talks about each of the albums. And you can see how her music changes over the years and how she's matured over the years. And she talks about life experiences. And if you think about our career, and now as a chair, I talk to my faculty in this way. I say, when you finish your training, I tell my junior faculty, it's the era of you. It's the era of self. You have to go and build your practice. You have to write for the grants. You have to do the experiments. And as you can see in the beginning, that's what we did in my lab. We were testing those reagents ourselves. But then in the next era, I challenged them to become the era of team. And it's there that you can make a tremendous impact. That's where you magnify what you do. And as I showed you in this slide, the whole brain tumor immunotherapy program was filled with people from all disciplines of neuro-oncology and even outside neuro-oncology. And so that, I see a lot of my mid-level faculty, and that's what I challenge them to say. That's how we're going to make a difference in the world. Because at the end of the day, the patients with glioblastoma, they want you to find a cure. And when you work together as a team, you may not always be the first author, you may not always be the senior author, but this is where we have a chance to make a difference. And then I say the last era is the era of giving back. And at some point, you have to realize that just like here, all these great people, the administrators, the leaders of this institution are giving back to the students to make sure that the students who really, I believe in my mind, are the ones that have the cure to glioblastoma, the cure to pancreatic cancer, the cure to restoring function. That's all in your mind. You have to give back, and you have to make sure that that generation succeeds. And that's the era of giving back. And in 2019, when I was about 13 years into my career at Hopkins, I had a busy clinical practice. We were having the clinical trials going. I had the opportunity to go back to Stanford to be chair. And it was there that my focus changed. While I'm still doing neuro-oncology, and that's still my passion, I got to take over a department of 58 faculty in neurosurgery, 32 of whom are the clinicians or clinician scientists, and 26 are scientists. And 16 of them have primary appointments in our department. And I get to oversee 21 neurosurgery residents. And when we did this in the era of giving back, we had to figure out our North Star. And like Dr. Usta said earlier, he has a clear vision of what this university is, where this university is going. We needed to have a vision of what we wanted. And this is the vision I wanted for us, was to precisely cure neurological disease while restoring function and well-being, to cure and restore. And I think this, especially for the students, this is your generation. Your generation will be able to find cures and restore function and well-being to people. We have our mission, which is to provide outstanding patient care. We innovate through basic translational clinical research, and we're very dedicated to developing the next generation of neurosurgeons, scientists, and leaders. But it's to cure and restore. So as an example, this is just work. This is not my work. This is Jamie Henderson's work and Krishna Shenoy's work at Stanford. This is an example of what Jamie and Krishna's passion was. So if you can imagine, someone who has spinal cord injury cannot move their arm because the spine is injured. So they said, well, can we figure out a way to give function back to people without going through the spinal cord? So what they did was they started recording signals from the brain, and they can put a little chip on the brain. And they started recording the signals, and through big data, they were able to interpret what the brain wanted to do. And so in that way, they were able to decode the brain and give some function back to people. So one of the first things they did, this was years before I got there, is that you can see that there's a little decoder. That's what the electrode looks like that's sitting in the brain. And this lady is connected to a decoder in the computer. And so the first thing that they did for this lady who was a quadriplegic, who couldn't move her arms and legs, was they gave this woman the ability to surf the internet. And as you can see, she's just thinking this and surfing the internet and looking at her favorite things, which are flowers. Okay. Now, fast forward about 10 years. And what they did was they said, we can do better than that. And we can give speech back to patients. And so this is really soft. So you all have to listen closely to this video. What they did was they took a patient with ALS who could not speak. She lost her ability to speak. And they went and figured out how to give speech back to this woman. And so you'll see the computer screen put up words, and then she'll grunt, and the computer will say the words that she's thinking, okay? So, oops, can you put on that video? Yes.
We'll start either way. Then you move to the next.
Yeah, the video's not there. This is the other video.
Oh, this is the other clip.
This is not.
You can move back, and
Can you play that video that you edited earlier?
Düzenlediğimiz videoyu oynatabilir miyiz?
No, not this one, but the other one. Do you remember you made that video edit?
Kestiğimiz bir video var diye onu istiyor.
I did well in school. I don't see much pollution turning on.
Our participant can also use the BCI to express herself. In this video, she's freely answering the question, what are you proud of?
I am a very good friend and sister.
Okay.
Finally, we also discovered that our BCI can
Okay, you can stop. Can we go back to the PowerPoint? So this was a paper they just published this year in Nature. And I remember when they showed this to me, it gave me chills because it was an amazing gift to give back to somebody. And as a result, I mean, it's an exciting time for people in the world today. And this is another video of a person who's a quadriplegic who always wanted to be a pilot, and he could fly a drone in the field, and he could fly a drone in the through an obstacle course. And so again, this was another gift to be able to restore function back to people. So I guess at the end of the day is what I tell people, like it's important to have a clear vision of what you want to do. And I think in this generation and for this particular era, we are in an exciting time because there's going to be a lot of great new therapies and great technologies that are available to you. And I was telling the students this morning, I do see in the next 10 to 20 years, people being able to write prescriptions to be able to cure cancer or give function back to people, and I think it's an exciting time. So to end this, I do want to acknowledge that, as I said, no one gets to this point alone, and I want to first acknowledge my family. I know that it's actually, there's close ties between Turkey and South Korea. As you know, Turkey sent troops over to South Korea to help during the Korean War, and in some ways, we're interconnected because it gave my parents freedom and the opportunity to come to the US. And my father came to the United States knowing no English and realized education was the way to go. And he memorized the majority of the dictionary just to get into college. And it wasn't a big fancy college, but he got a degree in engineering and worked as an engineer. And through the years, my dad unfortunately got laid off a lot, and they ended up having a convenience store. And despite having a convenience store, they worked in that convenience store for 22 years. And because of that, my sister and I could get an education and had opportunities in the US that they could only have dreamt about. But from them, I learned the skills or the values of hard work, resilience, and persistence. They've been told no so many times in their life, and they still kept moving forward. I also want to acknowledge my wife, who's my best friend and love of my life, and also allowed me to stay many late nights in the hospital. And my two daughters, Olivia and Violet. And then you can see my dog. I'm allergic to the dog, by the way. And my youngest one wants the dog, but I'm holding the dog. And if you notice, we're on a hike and the dog doesn't like to go on walks. So, but she's a great dog. But again, I want to acknowledge my family to say thank you. I also want to make one last comment about the future, and that's the students in this room. And I got to work with amazing students and medical students and postdocs through the years. And these are a list of people, and I'm very proud of them. They all got their first choices or second choices for residencies. They're on faculty now. But there's a really interesting study out there that shows that creativity is lost when you're by 40 years of age. And these folks were the ones who came up with a lot of the great ideas. How to, like, for example, Ayush and Chris, when he was younger, were the ones that came up with the mechanism idea. Just like in this room, I see the students here, you all have so many great ideas. I think that's the most valuable resource we have in this world, is the student and the futures, as well as our collaborators. I also want to acknowledge our clinical trials division that also helped the physicians. And then I want to acknowledge my relevant funding. So again, I want to say thank you for this award. I'm so incredibly honored to be here. I'm so impressed by what I've seen here today and over the past few days. And I think the future is very bright for you, and I think there's no limit to what this university can achieve. And congratulations to the students here, and thank you again for your time.
Professor, thank you very much for your speech. Now, we will continue with the photo shooting, so please stay here. Şimdi değerli mütevelli heyeti başkanımız Muharrem Usta'yı, sayın rektörümüzü, rektör yardımcılarımızı, sayın dekanımızı ve dekan yardımcılarımızı fotoğraf çekimi için sahneye davet ediyoruz. Manifesto dersi öğrencileri sol üst kapıdan çıkış yapabilir. Hocam ona geldiler. Geldiler hocam. Geldiler. Kıymetli öğretim üyelerimiz, profesörlerimiz, lütfen siz de burada kalın. Şimdi gelip fotoğrafımıza eşlik edebilirsiniz. Hocalarım, fotoğraf çekimi için sahneye davet ediyorum sizi. Buyurun. Biraz büyüyecek sanırım. Kamerayla. Ne çok şey oldu. Sizi engelleyecek. Kürsüyü alabilir miyiz? Kürsüyü alsak güzel olacak. Sadece biraz sağ almanız yeterli. Teşekkür ederim. Öğrencilerimiz fotoğraf çekmek isterlerse gelebilirler.